This protocol provides a general method to measure the endocytic rate of most plasma membrane proteins by using reducible membrane impermeant biotinylation reagents. In this procedure, cell surface proteins that contain extracellular lysines are covalently labeled at 4 degrees Celsius with sulfo-NHS-SS biotin, shown here in green. Next, cells are warmed to 37 degrees Celsius to permit endocytosis of surface proteins. Cells are then returned to 4 degrees Celsius to stop membrane trafficking, and residual surface biotin is stripped using a reducing agent that cleaves the disulfide bond. Thus, the only biotinylated proteins that remain are those that originated at the cell surface and were internalized and protected from the stripping procedure. Finally, cells are solubilized and biotinylated proteins are isolated by streptavidin affinity chromatography.
Hi, I'm Gabriel from Melikian's lab in the Department of Psychiatry at University of Massachusetts Medical School. Today we'll show you a procedure on how to measure endocytic or internalization rates using a reversible biotinylation technique. We use this procedure to measure the internalization rate of dopamine transporter. So let's get started.
One day in advance of the experiment, prepare cells and substrates for use on the following day. Cell types and media will vary, but if experimental cells are not strongly adherent, use an appropriate cell adhesion substrate, such as poly-D-lysine-coated tissue culture plates. On each 6-well plate, label 4 wells in total, using 2 separate plates for each protein tested. 2 wells on the control plate will be used for the total surface protein and stripping controls. Prepare a well on the experimental plate for each internalization condition being tested. Plate 1 times 10 to the 6 cells in 2 milliliters of cell culture